I'm Tom Rawson in the Mathematical Ecology Research Group at Oxford, and I'm in the final year now of my PhD, and it's been a PhD with the sort of blanket title of Mathematical Modelling Approaches towards Campylobacter Dynamics in Broiler Flocks. It's been supervised by Mike Bonzel and Marion Dawkins, and frankly Fran as well to a point by this point, just in a more unofficial format. And so the stuff I'm going to talk about today is primarily just summarising the first chapter of this thesis. And what I've done is a stochastic model that directly simulates strains of Campylobacter in a flock of chickens. Now, thank you very much everyone for coming, and I appreciate that often a lot of people are a bit nervous to attend a talk with mathematical modelling in the title, but I'm not going to have any equations in the talk. We're just going to talk about the sort of underlying dynamics at play and some of the results that we draw from that and how that might be relevant for you. I think that one of the reasons to display this is as a model, a model's only as effective as how it's used, right? And I think hopefully it could be helpful for people to try and either test hypotheses of their own in their own work. So I'll jump on in, and the benefit, I suppose, of talking to the Campy group is that I don't have to spend the first 3 minutes talking about what Campylobacter or chickens is, but I include the slides anyway for posterity, as I'm sure hopefully everyone is aware. Campylobacter is one of the leading causes of food poisoning in the UK, primarily gained through poultry flocks and poultry meat, and this has really overtaken the commercial poultry industry by storm. As we see this first column here, lots of our supermarket chickens are contaminated with Campylobacter, unfortunately. But one thing to take away again from these percentages is, 70, 80, 60% infection rate. That's not necessarily within a flock. What you tend to see when a flock comes into a slaughterhouse, we normally see that either a flock is mostly all clean or mostly all infected. There's a very few cases where it might be somewhere in the middle, but normally it's an either-or case. And the reason for that is that through the literature we see that once an infection of Campylobacter starts taking hold in a flock, it can fill the whole flock very rapidly, just about 2 weeks in total. So this is a graph I just pulled from one of the models from about 10 years ago now, for a few people that are just sort of following how many chickens in a flock get infected over time. And for the most part, up till maybe about 5 years ago, this was the best mathematical models we had, really, just some measure of how quickly a flock gets infected, which really does a disservice to the field to not be able to sort of, we've done all this fantastic research. I was quite surprised, actually, that we hadn't sort of pulled this together yet to get a better understanding of the underlying dynamics we see here. And it's especially a timely point in, I suppose, the Campylobacter history to get involved with this, because there's been so many studies now trialing various different biosecurity protocols, none of which seem to really be giving us the results we're hoping for. And so this is the point where we really want to turn to mathematical models. We've done all this work, we've got all this data. What can we actually learn from this, and how can that inform our next steps that we might take? So the way I do that is we consider in this model 4 basic variables, 4 quantities that we're going to be monitoring over time in this simulation to see how they evolve. And the first one of that will be the amount of Campylobacter in the gut of a chicken. Literally, within one chicken, how much of their total gut flora is made up of Campylobacter. Equally, we also want to measure how much of other bacteria there are. Campylobacter is not just by itself here. When it takes hold in a chicken, it is competing with other bacteria for resources here. So we also want to get a measure of how many of those bacteria there are. And then also, Campylobacter does elicit a host response as well, these host defense peptides. That's something else that we want to model in here. How does the chicken respond to Campylobacter and try and fight it itself? And then lastly, an important factor to capture here is maternal antibodies. So most chickens, when they first hatch, there'll be like a good 1 to 2 week period where they will very rarely show infection with Campylobacter, even when inoculated. And the reason we think this is that when a chicken first hatches, it has this pool of antibodies that it gains from its mother, and these wane over time. But initially, in early life, this really is strong at fighting off Campylobacter. So once you decide, okay, these are the things that I've seen in the literature, these 4 variables that we have information on and that I want to try and simulate, then it's about using the literature to work out how all of these 4 parameters for variables even interact. What are the rules that govern how they work with each other, right? But of course, one thing we can say as well is that the the microbiome is an incredibly sort of frenetic ecosystem. We have dozens of different bacterial species having some quite big impacts on what happens here. So you can either try and research every possible bacteria that you might find within a chicken and the exact way that it competes with Campylobacter. Or what you can do instead, if you want to try and simplify the amount of maths we've got here, is use a stochastic system instead, i.e. we introduce small perturbations to these variables, little random jumps up and down, which we'll see in a minute. And if that seems slightly perhaps cheating in some way, we've had some studies from zebrafish, which are skin thin enough that we can actually observe the microbiome in action by sort of dining certain bacteria so they show up in one microscope. And we actually show that stochastic models are really good at capturing the really frenetic stochastic dynamics that we see in there. And hopefully I'll show you as well where this model lines up to experimental data. So these 4 variables I talk about, what we do, we build these system of equations that describe the rules of how they interact, and then we can press go on a simulation, and we get something like this. So this is an evolution through time within one chicken by himself who's been inoculated with Campylobacter, and we see how that evolves over time. So the blue here is Campylobacter, and at the very first start experiment, we have that small inoculation, and with the purple here, these maternal antibodies. So what we see is those maternal antibodies really suppress that amount of Campylobacter, and they keep it really low and kicked out of the system so it can't take hold. But what we then see in this simulation is after those maternal antibodies wane and they run out, suddenly we create a niche where the Campylobacter that is still there, that tiny amount that didn't quite get destroyed, starts to replicate and take hold, and suddenly we have this huge explosion of the Campylobacter population as soon as it takes on in early life. But then what happens when you have a surge in Campylobacter? Then the yellow line comes in, those host defense peptides. The chicken's body responds to this, and it releases these host defense peptides to fight off the Campylobacter, as well as some competition effects from the other bacterial species. And we then have this sort of dancing stochastic jump around in the amount of the tug of war between Campylobacter and all the other bacterial species. So that's our basic dynamic profile. We have a sort of big suppression window, a sudden surge in the population, and then we bounce around in this different period. But this is specifically why it's useful to have these stochastic systems. So you can see all these little wiggles and jumps in the lines here. It's these little stochastic perturbations. Is those small perturbations can have huge impacts on actually the dynamics we see. And as anyone who's done some isolated experiments know, chickens aren't going to respond identically the same way to Campylobacter challenge. So if I press go on this entire simulation, the exact same model again, we might see something quite different. So if I press go again, maybe we see a case where after the maternal antibodies wane, that Campylobacter just stays a really low population for a long time until finally building back up in a huge way a lot later. Maybe if we press go again, we can see some cases where instead it has these huge jumps for a long time. The system, the chicken itself can't respond in a uniform manner to challenge. We see some really quite extreme spikes in Campylobacter. And of course, most importantly of all, when we press go again, we might see cases where Campylobacter doesn't take off at all, with those maternal antibodies actually just knock it out completely. And so we can test this out as well against the data we see in the field. So this is all chickens kept by themselves here. And I've got some great data from this great 98 paper by Akon et al., where they get a flock of chickens all kept isolated. They're all inoculated with Campylobacter at the point of hatch, much like the trials we've sort of just shown here. And we see a really good fit for what happens here. We see that, first of all, it seems like there's a slight dip in the population of how many birds are infected with Campylobacter. And this will be an artifact of those maternal antibodies getting them to such a low level that it's not showing up on swabs. Then we see that huge push where it takes off, and then slowly we get cases as well where chickens can end up clearing Campylobacter entirely. So that's chickens by themselves. And that's the model already working, just showing what happens in the system of one chicken. What we can then do is take this little system we've designed for one chicken, and we can extend that out to a whole flock. We can replicate these 4 equations, these 4 variables, X number of times for as many chickens as we want. And then what's interesting then is we can see how keeping loads of chickens housed together starts to affect the dynamics on each chicken. So what we're now going to do instead is show you some results from simulations of lots of chickens together. And the way we do that is we introduce a new variable representing the amount of Campylobacter in the environment, in the shared environment. So chickens will shed Campylobacter in their feces into the environment, and depending on how enriched that environment is with Campylobacter, the greater chance there is of chickens then ingesting Campylobacter and getting sick themselves. So I'm going to show you some graphs here where the bottom plot will be the Campylobacter in the environment, and on top we'll have 2 random chickens from this flock. Yeah, and see what happens here. And specifically, I'm going to vary the number of chickens that you might have in a flock. If I get myself a barn and I put just 2 chickens here, 2 chickens themselves, and I inoculate them both with some Campylobacter, this is what we see here. We see for chicken A, it has that first initial jump and then crashes out. That chicken is cleared of Campylobacter. The other chicken isn't quite so lucky and manages to sort of see these resurgences. And so hopefully you can see how this mirrors just these 2 chickens, makes up the entire flock, how that relates to the amount of Campylobacter in the environment. What I'm now going to do is cycle through some slides by increasing the number of chickens that we simulate in this flock. And each time we're going to start by inoculating only 2 chickens. So first of all, we up it to 5. And the first thing we're going to see is that our 2 random chickens that we take don't manage to clear the Campylobacter. And we see some slightly more bigger peaks in the amount of Campylobacter in the environment. And as I now just slowly increase the sample size, going to 10 chickens, 20 chickens, 40 chickens, 60 chickens, what we're starting to see now is that those peaks and troughs in the amount of Campylobacter in the environment are starting to get squashed down to this really uniform amount. And our chickens are getting more and more peaks. 80 chickens, 100 chickens, 150 chickens, 200 chickens. What's happening here? We have so many birds with an environment that is so enriched with Campylobacter, is our chickens are now being constantly having this endemic rate of Campylobacter just thrown at them at all rates. They're not going to be able to clear it, because even if we look at some of these graphs where, you know, the blue line here, the amount of Campylobacter might hit a really low amount, it doesn't matter because there's so much Campylobacter in the environment, they're picking it up again anyway. This sort of constant flat line here of blue in the environment, the Campylobacter in the environment, represents an endemic effect of Campylobacter being applied to the flock. I've got a few more of even more chickens as well. Yeah, we've got it all the way up to 400 here. But we can see, sure enough, chickens here aren't even reaching low levels anymore. There's just this constant amount of Campylobacter being thrown at them. And luckily, we can also check this dynamic against some flock studies as well from the literature. And sure enough, we can see that when we simulate exactly the experimental amounts, so what we do by that is we get the exact number of chickens that these experiments have in the flocks and the exact number of cedar birds. We see a really nice fit here to the red stars. This is just one experiment I've fit to of the exact point in time at which Campylobacter takes off and the rate, the attack rate that it takes over the flock as well. So already we're seeing just the impact of these large grouping so many birds together and how that affects the overall dynamics of the flock. The other benefit of this model, of course, is we don't just have to stick to one general strand of Campylobacter. Some of the most exciting work at the moment in the Campylobacter space is through looking at all the different sequence types and strains of Campylobacter available. And likewise, we can just replicate, we can make a new equation for different strains of Campylobacter. We can simulate multiple strains in the flock and see how different strains, different sequence types interact. So here, for example, that Campylobacter in the environment amount, I'm going to show you some plots instead of 5 different strains of Campylobacter. And if I showed you this plot, you might say, well, okay, well, it looks here like blue for whatever reason. It might be a weaker strain in some way. But actually, in all the plots I'm about to show you, these 5 strains have exactly identical parameters. They're the same equations I've just repeated 5 times for 5 different strains that we're going to monitor. And what we're going to see actually is these stochastic changes have a huge impact on the dynamic profiles of these strains of Campylobacter. So, for example, directly matching to this amount of Campylobacter in the environment, if I take 4 chickens at random from this particular simulation, we actually see this for their particular profiles. So the most striking thing here, right, is that although we saw that for the most part on the previous slide, most strains appear equally across the flock and are equally presented in the environment, when you look at chickens specifically by themselves, we normally see only 1 or maybe 2 sequence types that can really dominate the gut of that chicken. And that's something we're starting to see in genomics data as well from chickens on farms. And sure enough, if I then just stretch out the time span here, so we'd see how some chickens might evolve over time, we tend to see that every week or so, a chicken will have one strain dominant in their system that then gets knocked out and replaced by another one. And again, these 5 strains are completely identical demographically. I haven't made orange better than purple here or anything. It's just that different chickens will end up picking one at a time, and it's these small stochastic events which have a huge impact on what you see in the overall flock. Yeah. To get graphs like this, these dynamic profiles, what has to have happened is that at some point, this chicken B here has just happened to peck at the ground and pick up one part of the orange strain of Campylobacter, and that one event has now snowballed into this chicken being overly representative of the orange strain in its gut. And so then the very last thing that I use this model for is that we can use our model parameters and simulate it rapidly over time to work out which parameters in the model are the most important part for Campylobacter taking off. Which of all these parameters, all these dials and all the equations, which one, when I play with them, has the biggest impact on whether Campylobacter is going to take over the flock or not? And by simulating that loads of times, by using something called a Sobel index or a variance-based sensitivity metric, basically we get this number between 0 and 1, that's this STI. We get this number between 0 and 1, where the closer to 1, the more important that parameter is in causing Campylobacter infections in a flock, and the lower it is, the less important it is. So I've just picked some of the parameters out here to show you. So for example, we see that the decay rate of maternal antibodies, that's actually not that important. Although it has that initial squashing amount, it doesn't have a huge impact on whether a flock actually gets infected or not, which kind of makes sense when you think about it. It's important for that first 10 days maybe, but not for the farm for all the points beyond that. And what we instead see is that the most important parameters in the model are what's going on with all those other bacteria, all the other bacteria that Campylobacter is wrestling with for space and resources. Yeah. So what this would imply is that perhaps your best chance of keeping Campylobacter out of a flock is by somehow doing something to help just those other competing bacteria. And specifically, we also see that it's not too important about how much those other bacteria inhibit Campylobacter. You don't even have to enforce the bacteria that are shown to be good at, particularly good at inhibiting Campylobacter, like lactobacilli, for example. You just have to make sure there's lots of other stuff in there. That seems to be a really important way to possibly keep Campylobacter out. And so that's really where this model can be useful, is by looking for new ideas and essentially getting those sparks, testing out hypotheses you might have before you have to, put in a lengthy grant application. You can see, well, I've got this idea. Does it make dynamic sense by the mechanisms that we're seeing here? So for example, I might look at this and say, okay, perhaps I really want to look at doing an experiment here and working out what happens when I pump my chickens full of other bacteria somehow. So summarizing the main things I concluded in the paper that Desi kindly linked to be associated with this, the main things that I've just sort of talked about here is that as you increase the density of the flocks, the number of birds that you have in this environment and that are housed together, it really reduces the diversity and dynamic profiles between these birds. Remember those first simulations I showed where we saw really different graphs for each time we pressed go? The more chickens we put together, the less variation we see every time we press go. It starts to become far more predictable because we're getting the same amount of Campylobacter just forced on our chickens from the environment. The other thing we saw is that broilers, individual chickens, will only tend to usually express 1 or maybe 2 sequence types of Campylobacter at any given time. And the other thing, which I didn't talk about here, but that the paper talks about, is how that if you do make some of your sequence types or strains demographically weaker in some way, perhaps they don't replicate as quickly, a flock's really good at actually selecting and forcing them out quite quickly. The sensitivity analysis we put at the end there pointed out that competitive exclusion seemed to be a key driver in suppressing Campylobacter within a flock. And that most importantly, those dominant strains, the ones that we see the most often, doesn't necessarily mean that these strains are somehow demographically better in some way, some phenotypical advantage. It can be entirely down to just the random chance, not necessarily survival of the fittest strain, but just survival of the strain that got there first, the right chicken at the right time. And so one thing that hopefully the reason I wanted to talk about this model, and perhaps it might be helpful for anyone listening to this, is perhaps you've got an idea from your research, something that you want to test out itself. So when I gave this talk at a welfare conference back in the pre-COVID times, one of the key questions there was, how important is housing density? And I actually came in to the PhD thinking, well, it seems obvious to me, right? Just give these chickens more space. It's obvious, right? Then it'll go away. It should be no problem there. We can use the model to simulate that and say, well, what happens when we do give chickens more space? And so all I did in that is I just increased that environment variable, the total amount available there. And what I saw is that as I increased the space available, it actually took quite a lot more space to really see an impact on the dynamics. Yeah. So, for example, if I gave 100 times more space to a flock of chickens, it only reduced the probability of a flock infection from 100% to 94% likely. And to even make it just more likely for a flock to not get infected, you've got to give them 250 times more space. That is an extraordinary amount more area that just isn't viable, really. So me coming in initially thinking, well, this could be a good experiment to do. The model now shows perhaps there isn't much actual mathematical evidence behind this having a huge impact, and we might have to think of something else instead. So basically, that's the end of the main point I want to talk about from the model. And I'd urge at any point, if you do have, if anything that you'd heard here has sparked an idea off in your head and you think, well, I'd like to use the model to test this out, but maybe you're not quite so mathematically savvy, please do get in contact at any point and we can query the model and poke it around and see what it does. There's my email at the top there, and there's also the reference for this paper as well if you want to jump into the equations yourself. And yeah, I suppose this model has then sort of shaped the direction of the rest of my PhD. So some of those, again, those ideas I talked about in the previous slides of poking around, we had this idea that maybe to look at bird differences instead. So something that Marion, Fran and I did more recently on a more recent paper was some great data that Fran had on looking at the individual strains within flocks. And sure enough, we saw that these strains, as our model previously predicted, it wasn't necessarily that they were better in some case, but it was the bird differences that were driving this. We were seeing that actually but some birds were more susceptible to infection, that they were the ones that were really boosting certain strains to appear. But now I'm rambling anyway. I will turn it back now to if anyone had any questions. That's been quite enough talking for now.